welcome to another edition of Hammy Science. I'm Constable Ham, and this is my friend Hamstring. Today, we are going to talk about the world of developmental biology and a special gene named NFI. We will be using some big words, but don't worry, boys and girls. Constable Ham and Mr. Hamstring will help explain the meanings along the way.
We will also talk a little bit about our mouse cousins and the horrible experiment.
Whoa! Now these are kids we're talking to.
Oh yeah! Phew! That was a close one. Well, kids, I would like to introduce you to the wonderful world of myogenesis. Myogenesis is how our muscles grow, and the process occurs in stages that involve expression of different genes. Recently, research shows that Nfix, a special enzyme and specific member of the NFI family, regulates the transition between the embryonic stage and the fetal stage by repressing expression of embryonic genes while activating fetal genes and developing skeletal muscles.
Neato! I heard that the embryonic stage contains primary muscle fibers, while the fetal stage contains secondary muscle fibers. Is this true, Mr. Hamstring?
That's absolutely right. The cells that form during fetal myogenesis are very important for postnatal growth and regeneration.
Well then. Hey, Mr. Hamstring, did you realize that a lot of work went into finding out about Nfix?
Really? What kind of work?
Well, first, scientists had to determine if Nfix was even important in muscle development.
Tell me more. It sounds really interesting.
After scientists had a hunch that Nfix was important, they performed loss-of-function experiments on fetal myoblasts and gain-of-function in embryonic myoblasts. Loss-of-function experiments remove Nfix, while gain-of-function experiments add Nfix to experimentally see what happens. They transduced the cells with a lentiviral, which means that the scientists infected the cells with a virus that would incorporate its DNA into the cells. NFI-ENGR was a special DNA code that was incorporated into the virus DNA that shuts off the function of NFI proteins when transcribed and translated in the host cell.
Oh, I see.
Yep, pretty smart of them. They were able to analyze the proteins in the cell that were produced after the loss of function and gain of function experiments by first isolating the myotubes produced using a small treatment of a chemical called trypsin, and then using the SDS page and Western blot technique.
Sadly.
The scientists found out that MCK and beta-enolase, which are fetal protein markers, are expressed best only when the NFI family is not being suppressed from loss-of-function experiments or is being activated by gain-of-function experiments, while the exact opposite is true for slow myosin heavy chains.
Hey, I thought we were talking about Nfix, not the entire NFI family. How important is Nfix anyway?
Well, silencing experiments using silencing RNAs and real-time PCR show that Nfix, specifically, not the entire NFI family, was responsible for the activation of 68 genes in embryonic myoblasts and inhibition in 43 fetal genes.
Nfix sounds like a one-molecule show.
Oh, but I'm not! Nfix-2, an isomer of Nfix used in scientific research. Proteins, MEF2A and PKC-theta, are my good buddies. Separately, we are molecules, but together, we are a complex. As a complex, we activate MCK, an important fetal protein in fetal myoblasts.
Huh! I want to see proof of that.
No problem. Scientists can show you that co-immunoprecipitating the endogenous NFI in C2-C12 cells shows NFI is able to specifically bind MEF2A, but not MEF2C. Additionally, MCK wasn't produced in JEG3 cells that merely express MEF2A and PKC-theta without me. The molecular friends needed me to be their molecular bridge.
You are so cool, Nfix-2. Why do you need PKC-theta and MEF2A anyway?
Hey, don't be dissing my friends. Using chromatin immunoprecipitation, also known as CHIP, scientists realized that the PKC-theta is needed to activate MEF2A to its full potential during fetal myogenesis. However, of course, they also realized using CHIP that I was the key factor. I bind MCK and beta-enolase, however, along with PKC-theta, so that PKC-theta can do his phosphorylation job on MEF2A at the promoters. Then MEF2A activates production of MCK and/or beta-enolase.
Amazing! The super trio like no other.
Indeed we are. I don't know, Mr. Hamstring. All this talk about discovering that Nfix regulates the transition from embryonic myogenesis to fetal myogenesis through the use of activation and inhibition doesn't seem like real life, especially when Nfix 2 just told us that he helps activate MCK in fetal myoblast with his friends MEF2A and PKC theta.
What do you mean, Constable Ham?
I mean, isn't this only in vitro?
Gasp! Haven't you heard of the in vivo experiments too?
No way! Tell me about it.
Well, they crossed mice with special mutations. Specifically, they crossed flock N6C positive mice with MyoD CRE transgenic mice to produce mice that were Nfix deficient, only in developing muscle cells.
Huh. Tell me more.
The scientists found out that mice that are Nfix deficient are indistinguishable from wild-type mice as embryos, but have smaller muscles than wild-type when they reach the fetal stage. In addition, through the use of staining hind limbs with a chemical combination called H&E, using transmission electron microscopy, and performing Western blot analysis, the scientists discovered that the sarcomeres of the Nfix deficient fetuses were disorganized. Apparently, proper Nfix expression is crucial for sarcomere development.
Well, I'm convinced. I guess this is real-life stuff.
Hey, don't forget about good old slow mice and heavy chain.
Well, if it isn't slow HC, what are you here to talk about?
Well, for starters, I don't discriminate against embryonic proteins just because Nfix2 doesn't have time for us anymore. Why, when scientists use chip assay with Nfix2, they found that he doesn't even have time to directly inhibit my gene. He has to get with NFAT-C4 to downregulate me. When it was just NFAT-C4, my embryonic genes would just be racing around. Fibroblasts had robust expression of slow, myosin heavy chain. But then, Nfix2 and NFAT-C4 promoter became friends, and now I'm not even activated by because NFAT-C4 is all repressed.
Well, geez. I'm sorry. I'm just doing my job. I match up with the promoter and repress the embryonic genes, while at the same time I'm an activator for fetal genes. It's just what I do.
Well, I don't care for indirect repression. Kids these days with their myogen who sit and sarcomere thingamajigs.
Wait, MHC. Pack 7, what is it? What's going on, boy?
You require for myogenesis?
When switched on in a cell line, you cause a measurable increase in Nfix2 expression, and that in an Nfix chip assay, there were 4 binding sites for Pack 7 on the Nfix promoter?
Isn't that in a cell line, where you turn off Nfix2 is only minimally decreased, pointing towards Pack 7 being sufficient but not required for Nfix2? Expression and regulation of Nfix2 is almost completely unknown.
Well, what is it? What is it? Oh, you just have to go outside. Let me get the door.
Phew! Today has been a busy day. Together we've discovered the importance of Nfix and myogenesis.
Definitely. What's amazing is that scientists are already looking towards the future. For example, the time of expression for Nfix is quite important in different ways for different species. Future research will likely involve looking deeper into this exciting research topic.
Oh yeah, and almost nothing is known about their regulation. Therefore, examination of the gene pathway requires further study in the future.
Hey kids, if you'd like to learn more, go to these websites or look up these sources.